ONL Therapeutics is an emerging biotherapeutics company dedicated to protecting the vision of patients with retinal disease. We're developing first-in-class inhibitors of fast signaling, and ONL 1204 is our lead candidate entering the clinic later this year. We're well-funded to complete our preclinical activities, but we are raising capital to support our early clinical development. I'm privileged to work with a very talented management team and world-class scientific advisory board, who I'm sure you will recognize. And before I go too far, I'd like to just talk a little bit about the science behind our target FAS and its relevance to our lead indications. Disease pathophysiology and cellular stress in a wide range of retinal diseases upregulates the FAS receptor at the cell surface. FAS is best known for its role in apoptosis, but FAS has the ability to activate multiple death signaling pathways. In addition to apoptosis, FAS can activate necroptosis, think RIP kinases, pyroptosis, think the inflammasome, NLRP3, IL-1 beta, IL-18. For decades, our strategy has been to target specific intracellular targets of these pathways. But there are two key problems with that strategy. One is these pathways are highly redundant and there's significant crosstalk, and you end up with a whack-a-mole effect. You hit one target, you upregulate another. And second, by targeting these intracellular targets, we ignore the extracellular space, which is where we think much of the action takes place. In contrast, ONL's FAS inhibitors block all of these pathways and prevent and mute the immune response in the extracellular space. In fact, ONL has shown, and others have shown, that when you inhibit FAS, you downregulate all of these signaling targets downstream, including putative drug targets in red. So people in this room know well that patients with retinal detachment continue to have continuing unmet needs, and in spite of surgical success that many of you perform, visual outcomes remain poor. We intend to use ONL 1204 to treat patients at the time of diagnosis to shut down fast signaling until surgery can occur and the pathway is downregulated. This is an attractive indication from a clinical development perspective because we get a rapid readout in 6 months from that one injection. And it's also attractive because this is an acute anatomic event in relatively healthy eyes. And we can mimic this event much more accurately in models than other diseases. In fact, my colleague David Zaks and other colleagues at University of Michigan recently published the similarity between key markers in retinal detachment patients and the vitreous of retinal detachment patients with expression in the rodent detachment model. And I know this is an outer retina meeting, but if you'll indulge me, we are finding that injury to the inner retina is not all that different. And in fact, acute primary angle closure provides a unique parallel to retinal detachment, an acute anatomic event in relatively healthy patients that even with the best care often have poor visual outcomes. It is also an event that we can then model more accurately in our animal models. And again, here I'm showing you key markers, some of the same markers from retinal detachment patients in the vitreous of acute angle closure patients compared with other glaucomas and age-matched controls. And here we see these same genes upregulated in an acute glaucoma model. So let me quickly show you some data from 1204. I've previously shown this group that ONL 1204 is an exquisite inhibitor of photoreceptor cell death in a rodent detachment model. I've also shown that 1204 robustly protects retinal ganglion cells and their axons in an acute glaucoma model. And importantly, even when we delay treatment for 7 days after injury, after IOP has already peaked and is going down, we see that the protection is just as good. That really tells us that there is a treatment window opportunity after the acute attack has been broken. And it also tells us that it's not the acute injury in these diseases that is what's killing cells and leading to vision loss, it's the response to injury. And we see this really clearly when we look at the transcription data at the same time point. Here are the 3 key markers we identified in retinal detachment in our glaucoma model, and we see that ONL 1204 completely downregulates them. Here we see TNF-alpha, completely downregulated by ONL 1204. For our inflammasome fans in the room, completely downregulated by ONL 1204. And for our complement fans, completely downregulated by 1204. So I hope you can understand why we're so excited to be taking 1204 into the clinic. Our mission has always been very focused on patients, and we have never been more confident that FAST is the right target, that 1204 is the right molecule to bring new and important benefits to patients with retinal disease. Thank you.